important questions are our ability to understand how organisms are actually able to grow and how they're able to divide, the dynamics of the process, and of course, if we're thinking about infectious agents, how we can inhibit those processes as well. So the history behind the project comes from the strengths that we already have in Sheffield, particularly across the disciplines of physics, chemistry, through to biology and medicine. So we already have a strong foundation, and that's why this project is being taken forward, because it's to bring together the synergies between those different disciplines to allow us to succeed. Well, a big challenge, particularly in microscopy, is resolution, our ability to see very small objects. And until relatively recently, a big limitation has been able to get through is something that's called the diffraction limit. And now there are various technologies that have been developed which allow us to get round this problem and take it to the next level. So really we're beginning to see things which have never been seen before, allowing you to ask scientific questions which previously you couldn't address.
It's really quite fundamental in terms of the understanding that we want to get of how bacteria work, how they work really at the mechanistic level. But then, we're going to apply that to understanding the problems of those difficult bacteria that cause us problems.
We're bringing together people from different directions, from different backgrounds, so from physics, from biology, from chemistry, and we're using those different people to try and solve problems. So by being involved in a group working together like that, there's an opportunity to bring in your individual expertise and really make a difference to something that's bigger.
An organism like MRSA, Staphylococcus aureus, that is very small. The current imaging technologies up to now have only really been able to show where molecules are inside the cell, but not to be able to localize them and not to be able to look at their dynamics. The new technologies will allow us to do that, to gain an understanding of what underpins the life of that organism. If you understand the life of the organism, then you have the capabilities of actually determining and developing new drugs which are able to inhibit those life-giving processes, and therefore you've got new antibiotics.
Many people, I think, will have been touched by the problems of people being infected with these drug-resistant bacteria that cause terrible trouble in hospital-related infections in particular. So it's a growing problem, and we think that the kind of fundamental understanding that we'll get through this project will make a real big contribution to helping solve that sort of problem. The university is really strongly backing this project because we're very excited by this vision of combining the biological drivers to try and understand the mechanisms of biology at the molecular level with the development of the techniques that will make that possible.
So in order to facilitate this, what we need is equipment. And we've already been successful in gaining grants from the research councils, from the Wellcome Trust, from the Wolfson Foundation in order to buy equipment, but also to be able to build the facilities which are necessary for this project. So there are 3 building programs. One is a light microscopy facility for optical microscopy. One is a new EM suite which will be associated with the new state-of-the-art electron microscope. And the third is a biophysical imaging centre which will be in physics.
We've got the physicists and the biologists working together so that the biologists are really steering the direction in which the physicists are trying to develop the techniques. So in that way, we're really getting that link between what people want the techniques for and the development of the techniques that I think will make us, make the developments go much faster.
So imagine aims to make University of Sheffield at the forefront in this area, really taking forward these new technologies and applying them to important questions in biology and medicine. So this is a very exciting time for microscopy, and I think in the next few years, for us, it's most important to be part of that process.